So I would like to first thank the organizers for letting me to showcase advanced analytics at Roche. I'm going to skip explaining that artificial intelligence is really creating a revolution in healthcare, because I'm sure you heard it before. I'm going to focus on a very specific case where we apply advanced analytics. And this is automating tumor burden in assessments of drug effectiveness in oncology. So at Roche, we collect diverse kinds of data. And here today, I'm going to talk about the clinical trial data. And this data is used by the advanced analytics techniques to improve basically the life quality of patients through their journey. And there are many ways in which the advanced analytics contributes to this. I'm going to focus on accelerating the discovery and development of new drugs. So because we're such visual creatures, it's actually very important for us to actually see that a new drug is diminishing the size of the tumors that cancer produces. And therefore, there is a very important metric in drug effectiveness assessment. The metric is called CT-RECIST, and it stands for Computed Tomography Response Evaluation Criteria in Solid Tumors. It's an FDA-accepted metric, and it's de facto a radiological standard to quantify how effective the treatment is. Maybe I should say a few words about the importance of assessment of the drug effectiveness. So you might think that in developing a new drug, the main thing is to actually find a new molecule which works. It turns out that proving to ourselves and to regulatory agencies that it really does work is every bit as time-consuming and costly as actually finding the molecule itself. So this is a very important problem. If we could somehow accelerate this step, we could help a lot of people who use our drugs. So let me say a few words about the specifics of this metric. So first of all, it only works on solid tumors. It's something you can measure, but most of cancer cases are like that. And what radiologist has to do right now is to find up to 5 target lesions. And here we can see there is one lesion visible. So it's up to 5 such lesions, and all he needs to do is to measure the largest dimension of each lesion and to add up these numbers. That's the metric. It's very straightforward, but I would say very simplistic too. The only constraint on the radiologist is that there should be at most 2 lesions per organ. This is actually important because for us it means that not only we have to find lesions, but we have to find which organs they belong to. Sometimes it's harder to actually find organs for a computer algorithm than to find lesions. There are also up to 10 non-target lesions, but these are just auxiliary, so they're less important. So here I show that you have to find the lesion, you have to find the organ to which the lesion belongs, and finally you have to measure it, and then you add up all these measures. And finally, of course, once you get this number, you have to follow through different time points and see what happens to this metric. So if it is decreasing, it's a good thing. If it's increasing, it's a bad thing. If it doesn't change, it means the drug is just not doing anything. And here I show that it decreased, probably enough to classify this as a complete responder, which means that for this particular patient, the drug was very efficient. So our aim in this project is to develop a fully automated CT-RECIST algorithm. And I'll explain why it is important. So there are many ways it can improve the path which drug takes through this pipeline. And the obvious one which we can contribute to is just going to be faster and cheaper to develop a new drug. But the truth is that we hope we can do better than that, because The problem is that right now, all that the radiologist is estimating is the size of the tumors, the total burden. And that's, as I said, it's very straightforward, but also a very simplistic metric. The problem is that suppose you produce a new molecule and you test it in a large clinical trial, and you find out it does nothing. It does not necessarily mean that the molecule is not good. It might mean that you didn't find the right patients for it, because we know that even the same type of cancer, it develops differently in people with different genetics. So maybe by being more careful about what kind of tumor this particular patient have, we could find better patients for this particular molecule. And instead of just throwing it out after many years of research, we could actually use it for a smaller population, but it would still work for them. So it would benefit both the industry and the patients. All right, so the automation tasks for this complete automation of the CT resist include lesion-inclusive organ segmentation. It's very important and very difficult to segment organs which have large lesions because they don't look like normal organs, but they have to be included. Otherwise, we miss to which organ it belongs. Then we have to find the actual lesions. Then we have to register these lesions between different time points so we can actually trace what's going on with them. And once we get this registration, we have to come up with a classification of the progression of the disease. It could be a complete recovery, a stable disease, or something else. So in this talk, I'm going to focus on just the lesion segmentation part. As I said, organ segmentation is actually sometimes harder because lesions are something abnormal, and we as humans are actually quite good finding abnormalities among normal things. Organs are normal, so it's actually harder to segment them for a computer algorithm because there is nothing which stands out, basically. Okay, but I'm going to talk about the lesion segmentation. First, I'll just say a few words about why it's so difficult for a computer algorithm to find lesions. Well, one thing that most of the patients which take part in clinical trials, they come with an advanced stage, which means that they have sometimes hundreds of tumors, and a lot of them are very small. And to train an algorithm well, you don't want it to miss even small tumors, because other patients come with very few tumors and they're very small, so we don't want to miss them in these patients. So we basically have to find all the tumors no matter how small they are. Another problem is that human radiologists actually rarely agree on the tumor burden. So the scatter plot shows the metric, CT resist, compared between pairs of radiologists. So each dot represents a scan, a CT scan, and it represents like how two different radiologists estimated the metric. And you can see there's a humongous scatter here. So it's a very subjective metric. And why is it so subjective? Well, you can kind of get a hint here. So on the right, I show the actual tumors, all of them, which exist in this patient. And on the left, there are the 3 tumors that a particular radiologist chose as the target tumors. So he doesn't have to choose 5, he can choose just 3 for the CT resist. So you can see that even just choosing which tumors are target and which are non-target, it's already quite ambiguous. And then on top of that, he has to measure the largest dimension for each tumor and to add them together. So we hope that simply automating this, we can come up with a more objective metric. But again, as I said before, it's probably more important that because we can do this so much faster than a human radiologist, we can do a lot more than simply measuring the size of the tumor. We can measure the shape, we can measure the texture on a CT scan, we can measure contrast, we can do a lot more, and hopefully these different measures, they will contribute to understanding what genetic variation of cancer this particular patient has, and whether this patient is a good fit for this molecule or not. All right, so I'm going to talk about 2 algorithms that we are testing. They're not finished projects, so I will just show just preliminary data for both of them. So the first algorithm is more like a traditional machine learning or deep learning approach, and it's based on training a deep learning convolutional network with human-labeled data, which means that we need at least several hundred of CT scans of the full torso, where every single tumor is segmented by human. And I just showed you an example in the previous slide. And you can imagine that can be a very long process to collect this data. But nevertheless, it can be done. It just takes a long time to wait. And we have some preliminary data for this algorithm. So it's a two-stage algorithm because CT scans, they're large chunks of data. So it's very hard to, well, it's impossible to fit the whole CT scan at full resolution into, let's say, a three-dimensional neural net. So people have to come up with some ways to handle these memory limitations. So we used a two-stage approach. In the first stage, we just find the ROIs, which are the most likely regions in the whole torso to have a tumor. And then in the second stage, each ROI is scanned by the second algorithm to find lesions inside the ROI. This way, it's much more memory efficient. This network is also interesting because it's a probabilistic network, which means that it can produce slightly different lesions every time you run it. And that's very useful because, as I showed you before, when you collect human segmented data from human radiologists, you can actually have different segmentations for the same CT scan. And you want to reflect this natural subjective variability in your model. This is why we use the probabilistic model. So here you can see in green the human labels, in blue the model labels. And you can tell that there's a pretty good agreement. Just to put some numbers in it. At the ROI level, the sensitivity is 94%, which means that 94% of ROIs defined by human radiologists and by the model, they overlap. And the dice score, which is the overlap of the 3D segmented lesions, is 82%, which is also not bad, especially given that we still don't have these hundreds of CT scans to work on, as I mentioned. So we're still waiting for more data to train this model. So this looks pretty promising. The problem, again, is what I mentioned before, that manually labeling these images, it's very time-consuming and very expensive. So again, we have to segment all the lesions, because if we just segment the target lesions, which is much easier, then we're going to leave a lot of smaller lesions unsegmented, and that will definitely result in a lot of false negatives. And as I said before, there are sometimes hundreds of these lesions, so it can take up to several hours. I say an hour, but actually it can take up to 3 or 4 hours of the radiologist's time to get all these lesions just to define the ROIs for them. And then it takes 3-4 times longer for an imaging specialist to actually outline the lesion in every slice. So as you can imagine, it takes thousands of work hours and millions of dollars to get this kind of data set. So the question is, can we do without it? Can we find an algorithm which doesn't need human segmented data? And that's like the latest development. And if we look at how well humans do that, we actually see that we can do it, because, for example, I can show you these two data sets. I don't expect you to be an EEG specialist. And nevertheless, if you look at the left set of traces, which are supposed to be normal EEG traces, and you look at the right set of traces, you will immediately start noticing the difference between what's supposed to be normal and what's supposed to be abnormal, which is a seizure. So after a while, you will probably come up with these episodes as abnormal. So if we could do it, then probably a deep learning algorithm could do it too. Why not? So importantly, these traces, they're unpaired, which means that, like, for every trace here, there is no trace like this one, but with abnormalities. They're completely different. They came from different patients in this case. The same thing with CT images. We can actually find abnormalities in CT images. I mean, the algorithms can do it. even if CT images are impaired, which is of course the case most of the time. You cannot expect to have a healthy CT and CT with tumors for the same patient. So what we could do is collect a large set of healthy CTs and a large set of CTs with tumors and train an algorithm to find the lesions, to find the tumors, with only the set level label, that this set has tumors, this set has no tumors. And that, of course, will save us a lot of time and money. And the results I'm going to show, they were based on 1,300 chest area CT volumes with cancer. It was all lung cancer. And 300 CT volumes without cancer. That was actually very hard to find. It's hard to find CTs of healthy patients. And just so that you're not surprised by the colors, it actually helps to cast certain values in the CT range to colors. So for example, muscles are cast to green and lung tissue CT values are cast to red. That actually helps the algorithm to work better. So these are the results. And I don't have time to explain the algorithm, but basically the way it works is that it takes the original CTs shown here, and you can see some tumors in it. And then what the algorithm does, it actually removes them. It creates analogs of the same CT. It creates a pair, how this CT would look without tumors if it was from a healthy person. And that's the second column. And then, of course, to segment the tumors, all you have to do is to take the difference, and with some very simple morphological cleaning up, you get the tumors here. So far, so good. It looks quite promising. And that's all I had to say. So if you have more questions, you can come to the booth, or you can ask some questions now.